30 people, 25, 30 people joined already.
Yeah, I think we can start. Okay, so welcome everyone to join our bioinformatics and genomics seminar today. So Dr. Kuhn and I from the BCH will co-host this meeting. So Dr. Kuhn will introduce our speaker today. So before actually Dr. Kuhn introduce our speakers, I would like to share some good resource for the new audience. So we actually recorded all our seminars in YouTube. So if you are interested in any of our previous talk, so I just suggest you go to our RC Wiki page. So if you have a browser, then type RC Wiki, and it will go to our research computing wiki page, and then scroll down, go to the digital science education, and you can click the link here. So in this page, we actually put all the upcoming seminar and the previous seminar here. And if you are interested in any seminars or previous seminars, you can either click the link here or scroll down. You can find all the YouTube here. So if you are interested in any of these topics, you can save the image, save the videos and watch it later. With that, I will pass to Dr. Kuhn. So please, Dr. Kuhn, can you introduce?
Hi, I'm Saekwon Kong at the Computational Health Informatics program. Myself is a psychiatrist and a genome scientist, like what Dr. Michael Inouye is going to present today. It's my great pleasure to introduce Dr. Michael Inouye. So currently, Dr. Inouye is the director of the Cambridge Baker Systems Genomics Institute in Australia and the Turing Fellow at the Alan Turing Institute. And Mike finished the undergraduate study at the University of Washington in the biochem and the economics, and completed his PhD in the computational genomics at the Leiden University and the Sanger Institute. After his postdoctoral training at the WEHI in Australia, and he has been an associate professor at the University of Melbourne from 2012, where he founded the Center for the Systems Genomics. So he's a founding member and the director of that Center for the Systems Genomics. And then in 2017, Mike was recruited to the Baker Institute in Australia and set up his lab also in the University of Cambridge in the UK. So he's currently located, physically located at the University of Cambridge in UK because of the COVID situation. But he going back and forth to Australia and the UK and maintaining two sites, very active, productive two sites. And the Mike's research focuses including the systems genomics, the polygenic risk score, and integrative analysis of multi-omics data, and the development of analytical tools for the computational genomics. So today, I believe that Mike is going to talk about the Polygenic Score Catalog, and which is the open source database of the published polygenic scores. Without further ado, so Mike, so stage is yours.
Great. So thanks, Saekwon and Liang, and also to Shira for the invitation. Could I just check that everyone can sort of see my slides okay and hear me all right? Just give me a little nod.
Yep.
Yes.
We're good. Okay, great. I guess and thank you very much for that introduction. Today I'll be talking about the Polygenic Score Catalog, as well as giving a bit of a more broader view and introduction to polygenic scores as well. So for people who aren't really in the field, in the know, hopefully this gives a nice little sort of primer as well. Okay, so I'll assume that everyone sort of has like a pretty good background about the human genome and the Human Genome Project, and a lot of genome-wide association studies, which have kind of taken over a lot of human genetics for the past sort of 15 years or so now. So polygenic scores really have their root in the human genome and in projects like the International HapMap Consortium, which really started once the kind of, I guess, draft human genome was finally finished in 2003. So the point of the HapMap was really to to map the common variation across the human genome in terms of these things called single nucleotide polymorphisms, or SNPs, and to do that for a select few sort of diverse human populations as well. And that really set the stage for genome-wide association studies, which, as you've probably seen from a lot of the publications, have really sort of, in a very high density way, mapped the association of loci right across the genome with thousands of traits and disease risks and that sort of thing. So those that data and indeed those summary statistics, whether it's individual level genotype data and phenotype data or the GWAS summary statistics, have really set the stage to construct these things called polygenic scores. So polygenic scores really came onto the scene. So they've been in sort of animal research for a while, especially in livestock, but the methodologies thereof really came into the human world somewhere around 2015, maybe a little bit before. And really what polygenic scores are is, at its simplest, a weighted sum of genetic variants weighted by their corresponding, say, effect sizes in genome-wide association studies. And what that means is that it basically aggregates a lot of the genetic susceptibility, I guess, to a complex trait or to disease risk from right across the genome. And so you end up with one score, one number per individual, which gives you an idea of what sort of their risk of disease is, or what their propensity is for high versus low BMI, et cetera. So when you plot out things like polygenic scores, you can see that you get these distributions across a bunch of individuals, and you can use that in downstream modeling, things like risk prediction and classification analyses and that sort of thing. Okay, so polygenic scores, as you can imagine, being based on germline DNA, have both kind of advantages or positives, and they've also got some real challenges as well. So of course they're kind of non-invasive. You can spit into a tube. The technology to measure SNP variation across the genome, or these very sort of relatively easy to use SNP chips from Illumina or Affymetrix. It's a very robust technology, very reproducible. Because it's germline DNA, it can be calculated very early in life. That's not to say that the effects don't change throughout life, but that's to say you can sort of do this sort of thing once, anytime in the lifetime of the individual, and you get the same thing. It's a one-time sunken cost of, and that's an old number now. It's actually cheaper than, say, 30 or 40 pounds or whatever the sort of exchange rate is now. I'm American, I should know these sort of things. And from that data, you can basically calculate now hundreds, maybe even thousands of polygenic scores. So you get sort of quite a bit of bang for your buck in a way, or quite a bit of data, I suppose. So, and it really, one way to think about polygenic scores is that they're a foundation for other risk factors. They've been termed the first risk factor in the context of diseases. But there are some real major sort of challenges there. It's been well recognized that because, well, a lot of biomedical research, especially sort of cohort collections, tend to be in European ancestries, and therefore a lot of the genotyping and GWAS and all these sort of things sort of roll onto each other. And that means that polygenic scores exert a European ancestry bias in terms of performance. They tend to perform relatively well in European ancestries versus, say, African or South Asian ancestries. So that's a challenge. There's also the challenge of polygenic scores are really kind of what I say, horses for courses. You can't sort of make a broad statement about a polygenic score or polygenic scores. Each one has like a different power and a different sort of utility, a different sort of use case, depending on the disease and clinical context, and indeed what you'd actually do with that information. So all of the kind of parameters here are really quite different depending on what you're looking at. And again, I mentioned sort of interventions, therapeutics, that sort of thing. Some diseases have interventions, right? But some don't. So you really have to kind of think about whether the information is really useful to the clinician or the individual themselves as well. So that's a complicating factor that we're still sort of working through. So we've done quite a bit of research since since the early days of starting my lab in 2012. A lot of it sort of methodology based. We've been looking at various diseases and in what context, that sort of thing. So really there's been a long sort of tail of studies sort of setting the scene and sort of building up to where we are now. And really now it's very much still not necessarily huge leaps in methodologies, but we're still kind of getting a handle on what diseases polygenic scores do well in, what are the contexts, and how well do they need to perform in order to have some sort of clinical utility, right? And I mean, I guess you'll sort of see my terminology morph and evolve, I guess, because over time. I mean, we've called these things, genetic scores, genetic risk scores, genomic risk scores, and polygenic scores. So this sort of terminology even has morphed over time, but I think we've kind of settled on something at least now. Okay, so I've kind of chucked this slide in because it is BCU, and I think that there is kind of a valid use case, I think, for polygenic scores in sort of supporting some of the diagnostic pathways of a lot of diseases of infancy and early childhood. So I would draw your attention to some of the work that's been done in Exeter around type 1 diabetes, newborn screening for type 1 diabetes. So people like Richard Oram and Seth Sharp have put together polygenic scores, or in this case genomic risk scores, genetic risk scores, that sort of thing, similar sort of thing, that have AUCs of really in the low 90s, which in terms of classification accuracy is really quite high. So basically that can be interpreted as over 90% of the time you're actually classifying a case at a higher sort of risk than a non-case, which is pretty good just on its own. And when you combine that with autoantibody screening, it shows promise, especially relative to what some of the methods people use now, which is HLA typing. And it also appears to have some sort of potential in terms of differentiating type 1 versus type 2 diabetes, which, especially in adulthood, is important because it can reduce incorrect treatment, medical costs, and corresponding morbidity. So another use case for polygenic scores that has really come on the scene has been in celiac disease, another sort of autoimmune disease which has a very strong HLA signal. And here again, you can contrast polygenic scores which have AUCs of roughly 0.9 again, to things like I guess having an HLA DQ positive risk allele. So roughly that gives you an AUC of about 0.8, and using a genomic risk score such as the one that we designed in 2014 gives you an AUC of about 0.9. So you can see sort of how that might improve upon things, and it might guide how you do serological screening, how you might follow up individuals at high risk. Let's say they have a first-degree relative with celiac disease, and how you might minimize the gluten challenges and intestinal small intestine biopsies, which are all part of that. And one other sort of application that I would highlight is also in juvenile idiopathic arthritis, where, as some of you probably know, there really aren't a whole lot of molecular tools to help the clinician make the diagnosis. So kids who present at pediatric rheumatology clinics, they go through a very long sort of multi-month process of largely exclusion to try and secure a JIA diagnosis, and indeed a subtype diagnosis if possible. So we're relatively optimistic that some of the recent genomic risk scores that we've constructed may have some clinical utility there, still to be determined, but you can sort of read more about that on a preprint that we've got on bioRxiv from Rodrigo Canavas and colleagues. So one of the things that my lab has really been focusing on, so we've done some work in autoimmunity, that sort of thing, but we've been focusing a lot on cardiovascular disease, and we do that because, you know, this is a really kind of challenging case. So, you know, in a lot of autoimmune diseases, you've got a very strong HLA signal. You've got sometimes, you know, other really strong loci coming into play, but you know, for highly polygenic diseases, that is, these diseases whose genetic architectures, you've got this really long tail of small effects that were really sort of sprinkled around the genome. That is a real sort of prediction challenge, a real sort of challenge for constructing polygenic scores. But for a lot of diseases, you know, these are the really common ones like type 2 diabetes, you know, obesity, you know, cardiovascular or you know, coronary artery disease, et cetera. So, you know, if we want to tackle those diseases, we got to figure out how to handle, you know, a lot of small effects sprinkled around. And early on, in the early days, you know, there was a lot of studies coming out looking at, you know, really small numbers of SNPs, just the ones passing genome-wide significance from a GWAS, so 5 × 10⁻⁸ in terms of a p-value threshold. And, you know, there's a lot of sort of, I guess, inconsistent results. You can imagine that when you've got a fairly weak sort of instrument, that sometimes it's statistically significant, sometimes it's not. Sometimes you get, you know, just depending on various parameters of the cohort, it gives you different sort of results. But, you know, a lot of the groundwork was laid there by Basek Katharison and Samuli Rapati and co. And it's really just sort of been gathering steam as we've gone, you know, through the years, you know, through the 2010s. So I'll just sort of highlight some work that we contributed to this effort back in 2016 in European Heart Journal. I won't sort of walk through the construction, I guess, here and of the polygenic score. But really just to say that that this was one of the bigger ones at that stage, sort of 49,000 SNPs built on top of the Illumina Metabo chip. And it was externally validated in some pretty large cohorts at the time. So FINRISK, about 12,000 individuals, a subset of the Framingham, which wasn't part of some of the initial GWASs, and another familial hypercholesterolemia study called ARGOS, which I won't go over here, but I'm happy to discuss offline. So, you know, as we sort of, you know, use these polygenic scores, obviously, you know, you can, actually, sorry, I've got this, this background. Okay, so as we construct these sort of polygenic scores, we want to look at them relative to conventional risk factors. So one of the first things we did once we constructed our score was we compared it to both the individual conventional risk factors that comprise the Framingham risk score, so things like LDL or total cholesterol, systolic blood pressure, smoking, et cetera, and how to look in, you know, what's the correlation between this the polygenic score and the Framingham risk score and its components. And really, basically, there's very, very weak correlation. You know, R-squareds of about 0.3 or 0.003 or 0.3%. So, you know, and this was, you know, consistent with when we actually looked at a modeling-based framework and sort of looking at, you know, C indices and hazard ratios and that sort of thing. So, you know, here you can basically see on the right that when you plot out the hazard ratio per standard deviation of this polygenic score or genomic risk score, that you can add in, you know, the other sort of conventional risk factor scores and the individual conventional risk factors, including family history, and you get very little sort of jumping around of that effect size. It really sort of sticks there at around about 1.7, and then you can basically chuck in all of the risk factors, including family history together, and it really only goes down a little bit to about 1.6. So, you know, pretty much orthogonal information, which is interesting because, you know, it kind of means that that the GRS, whatever sort of disease variants it's capturing, is independent of those conventional risk factors, And in a prediction sense, it means that you've got something that you can add there on top of conventional risk factors. So we kind of went ahead and did that and looked at things like C indices and how much additional C index you can get by adding GRS, just sort of dropping in that variable into the risk equation and estimating a weight for it, and see what it adds on top of conventional risk factors and conventional risk scores like Framingham and the, at the time, ACC-AHA 2013 score. And we see sort of consistent patterns across both FinRisk and the Framingham Heart Study, where you add in the genomic risk score, it adds roughly as much as the Framingham or ACC-AHA, but really because they are orthogonal information, and really the maximum sort of gain in C index is when you combine those two sort of classes of risk factors together. Okay, so all of that is really kind of informative from a conventional, or from like a very traditional cardiovascular disease risk prediction framework, right? So they tend to look at these are linear models that are predicting usually 5-10 years in the future. So a lot of times they're predicting sort of future 10-year risk, that sort of thing, from baseline. And really, one of the sort of unique properties of a polygenic score, right, is that it's based on germline genetic variation, which, as I mentioned, can be calculated early in life. And that really means that it's really on a footing more similar to, say, sex, which also is kind of genetic, but is something that you kind of know, like you kind of know that men are at higher baseline risk than women, for example, for coronary artery disease. So, this is kind of more of the framework of you've got this kind of very early risk factor, right, which you should be able to measure early in life that hopefully will help you guide what sort of screening strategy you might take. So we started thinking about GRSs in terms of trajectories, so lifetime risk trajectories. And these are sort of one of the early papers that really sort of map that out. And what I'm showing here is cumulative risk. So these are basically inverted Kaplan-Meier curves and plotting out quintiles of GRS within Framingham, men versus women, and in terms of FinRisk as well, FinRisk on the left. And really what you see is that you see people in the highest quintile, the highest sort of 20% of the GRS, their risk curve is much higher than people in the lowest quintile, so the green curve, for example. And to the extent that and actually I should also add, we sort of see men versus women here, there's no sex there's no sex interaction here. So really we're just talking about shifts in the baseline risk. So that basically means that when you're talking about the red curve versus the green curve, it's really just sort of moving it, sort of translating it left or right between men and women. So here in, say, FinRisk, you can sort of see that, and you really start thinking about this in terms of, well, if let's say I cared about a threshold of 10% cumulative risk, right? And that was something I would do something about, right? You can sort of see that the age at which you reach 10% cumulative risk is 18 years younger for people, the 20%, and this is also a population level estimate, right? So 20% of the population roughly reaches that 10% cumulative risk threshold 18 years before the other 20%, the other end of the GRS distribution of the population. So on the green curve, right? So, this is quite a lot of time in which to do something about people who, as far as we can see, are at higher lifetime risk of coronary artery disease. And you can play sort of other sort of, you can stratify by different things with this, with GRS as well. You can stratify by current smokers versus non-smokers. So here we can see that, men who are in, say, the highest quintile of GRS, so these are the high risk individuals, so the red curve that I showed you before. This is now sort of looking at non-smokers on the blue curve there. And you can compare that to individuals who are in the most protected sort of 20% lowest risk quintile of the GRS and who are smokers. And actually, if you're in the highest 20% of risk, but you're a non-smoker, you're still at higher cumulative lifetime risk of CAD than individuals who are relatively genetically protected, but are smokers, right? So you can start sort of looking at what the relative contribution is of, lifestyle or behavioral aspects versus people's sort of background genetics. And one other interesting sort of observation here really is that, for women who are in the lowest 20% of GRS, their cumulative risk curve is barely taking off there, right, all the way up through age 75. So, there's some proportion of women who we may be able to predict who really may not have to worry very much about CAD for, the majority of their life. So what that might mean for screening strategies is still an open question, but that's the data. So this was really kind of nice, really cool. Back in 2016, this was still novel. And what we ended up doing with our collaborators in Finland, so Samuli Rapazzi, Veikko Salama and co, is they set up a study to actually see what if you actually deployed this 49K GRS, and gave this information to individuals alongside conventional risk factors, how might they change their behavior? Would they lose weight? Would they see the doctor more? And what might that mean for downstream risk of CAD and CVD? So what Samuli and Elizabeth Whitten did is they went ahead and well, they actually kind of thought, right, about, we actually don't really have a good risk communication way of a risk communication tool to actually, communicate this risk to the average person, right? So they started by designing a tool called the Cardio Compass, or Cardiokompassi in Finnish. And it's a kind of a cool tool where they plot out these survival curves like I was just showing you, and you're able to kind of translate and shift it up and down using your conventional risk factors. So if someone stopped smoking, they could see what that does to their risk curve. If they lost 20 pounds, if they got their blood pressure down 10 millimeters mercury, that sort of thing. You can sort of see how these sorts of lifestyle choices combine with their sex, their age, and their genetic risk in order to basically flatten the curve, to use a sort of in vogue saying. So this showed promise. It was presented, this is old now, but 2008 at ASHG. The results at ASHG, these results look promising, and we're just sort of waiting for really, I think, events to accumulate before we see to what extent people who are, say, at high genomic risk make the kind of lifestyle modifications or might go through different sort of treatment regimes as people who are at low genetic risk using this tool called Cardiokompass. So life goes on. That's great. That's a translational aspect which came out of the 2016 paper, and we're eagerly awaiting the results there. 2018, UK Biobank hit the scene. Many of you probably use UK Biobank in your sort of day-to-day research. We sort of came together, the UK Biobank Cardiometabolic CHD working group, and really thought that what we should do is try and see how a polygenic score performed in UK Biobank. And we also leveraged some methodological advances. I mentioned methodology before. We sort of devised this new approach called metascoring, which I'll just sort of briefly describe. So instead of, say, taking a weighted sum and summing up the per kind of the per SNP weights or log odds from a GWAS, and multiplying that by the allelic dosages and getting one number for an individual, what we're doing here is we're actually leveraging multiple polygenic scores for an individual. And we're actually weighting the scores rather than the individual SNPs. So you're basically bypassing, well, not necessarily bypassing, but you're not necessarily looking at variation at the linkage disequilibrium level, not at the allele frequency level. you're looking at the score level, and you're combining based on the scores. So I guess this approach we called metascoring, or score of scores. And this has become sort of more popular with approaches like MTAG, and a few other approaches have been coming out recently as well. But it's basically the same idea, right? Where you've got some sort of weighted average of scores instead of SNPs. So this appeared to work really well in UK Biobank, where we used a few kind of external scores. We used a very sort of small training set of like prevalent CAD cases, about 3,000 cases and non-cases from UK Biobank to estimate the per score weights. And then we looked at the meta-GRS that we calculated in terms of prediction of CAD in the rest of UK Biobank and alongside conventional risk factors for incident CAD. And I'll sort of mention that actually in this case the actual the weighting step of the metascoring didn't really do a whole lot in terms of increasing the per SD hazard ratio. It probably only bumped it up about from like 1.65 to 1.7 per SD, something like that. So in this setting, not that much, but still a little bit better than what you would do using an unweighted score. So one of the main things that came out of this, using the 500,000 individuals that are in UK Biobank, was that we were able to compare this meta-GRS to a lot of the conventional risk factors, and again sort of see that well, it's independent of the conventional risk factors, and that in terms of C index, if you look at individual models of each one of these risk factors, the genetic risk score, the meta-GRS, outperforms basically all of them, unless you're talking about, say, sex or age, right? So those are still by far and away the biggest sort of risk factors. And indeed, I guess, when you're talking about survival analysis, age is also, in many cases, time. It's actually time in some of the survival analyses that we did here. So again, we can sort of plot out these inverted Kaplan-Meier curves, the cumulative risk curves, and see, a similar sort of, very kind of smooth, and you can sort of see the 95% confidence intervals there, those little dashed lines, in terms of the, quintiles of polygenic score, or meta-GRS. And again, you see this difference in baseline risk between men and women, and again, the absence of a sex interaction. Okay, so that's great, and I think one of the things that we were very happy with was that we effectively basically replicated everything in UK Biobank in terms of the performance of a polygenic score, just on a much more massive level, and really one of the things that we've been thinking about doing is trying to take those additional steps in terms of how we look at diversity, as I mentioned, the European bias in polygenic scores, and B, try and at least within European populations, see what the potential clinical utility is of polygenic scores. So first things first, there is variation in terms of the effects of polygenic score, and that's still the case with this coronary artery disease meta-GRS. So if you look on this plot of the hazard ratio per ancestry standard deviation of the polygenic score, you see that that British whites and other sort of white self-report, well, this is the PC determined ethnic group, that you basically get what you would in terms of what you'd expect, basically, so 1.7 or so. But that you start seeing kind of an attenuation of the effect size as you start going further and further away from the European ancestries that dominate the underlying GWASs, right? So you go to South Asian individuals, about 7,500 in UK Biobank, and you see that sort of point estimate going down to about 1.55. And then if you go to Black or Black British ancestries, your hazard ratio is roughly about 1.25 or so. So you see this attenuation. And one of the things I think that many of you probably know Peter Kraft. It's worth mentioning that one of the things that Peter really pointed out, or that at least I saw on Twitter, inevitably, is that he also makes the point that baseline risks are also different. So while we are seeing an attenuated effect size in Black or Black British, or correspondingly, say, African Americans, we also need to look at higher baseline risks, that sort of thing as well, because overall those two things combined, you could actually be preventing a similar number of cases despite there being a less powerful instrument, right? So we do need to keep that in mind. That's something that hasn't been looked at in any detail yet. One of the other things, so it's very good to kind of allow people to go out and so we very much share our polygenic scores. NHGRI eMERGE picked this up, and group Iftikhar in Minnesota picked this up and looked at it in the eMERGE data sets, looking at Europeans, African ancestries, and Hispanic ancestries as well, and did a benchmarking of different polygenic scores for coronary artery disease. And really, I think one of the positives thing here, is that they did a benchmark, which is unfortunately far less common in the literature than it should be. And B, it really gives you a sense of the robustness and the relative sort of attenuation of polygenic scores as you move away from European ethnicities. So this is worth looking at. I think one of the things that surprised us was that at least the meta-GRS in European ancestries and Hispanic ancestries perform basically the same. So the hazard ratio per SD is about 1.53 in both ancestries. And then of course in African ancestries, you see even more attenuation, roughly what's in line with the Black and Black British of UK Biobank, where in eMERGE, in African American ancestries is about 1.72, which was relatively good in terms of coronary artery disease PRSs. Similarly, other groups have gone through and completely externally validated this in out of our hands and everything in say French Canadian populations as well. So I'll just sort of go through this real quickly. So this actually just came out. This came out in Nature Communications last year. I haven't updated the citation. We also designed a meta-GRS for ischemic stroke, which in terms of C-index hazard ratio outperform basically all the other conventional risk factors except for blood pressure, which is not surprising because blood pressure is a very strong predictor of stroke later in life. But the point here really was that we wanted both coronary artery disease and we wanted a stroke PRS in order to look at cardiovascular disease more broadly, or at least the two major components of CAD and stroke. So really, we set this up and this is currently in review. And really what we're hoping to do is explore what I was mentioning before, clinical utility in terms of screening, different strategies for screening, whether we're talking about targeted screening strategies, say going after individuals at intermediate risk of CVD and using PRS for those people, or using a blanket approach, right? PRSing everyone and assuming everyone's got that information and seeing what that does in terms of how that might affect C-index or numbers needed to treat, needed to screen in order to prevent one case, that sort of thing. So again, we're using UK Biobank here. We are looking at the conventional risk factors again, and we're also using some of the primary care data in the UK from the CPRD database to try and recalibrate the risk in UK Biobank, because, as many of you probably know, it's a relatively healthy cohort. So this is all very important as you kind of get sort of more closer to reality, I suppose, instead of the performance of a predictor in all our kind of lovely cohorts. What might it look like in a more realistic setting? And we coupled that with a statin intervention assuming a 20% relative risk reduction, that sort of thing. And we're still building up to a genome first approach. This is still looking at it in the context of conventional CVD prediction, but hopefully getting onto that soon. So the headline results are basically, when you're looking at CVD, this is a composite outcome. So you're looking at coronary artery disease, you're looking at stroke, et cetera. So you've got a much more powerful PRS for coronary artery disease than we do for stroke. This is definitely the case. So that when you roll those two outcomes together, you're looking at a bit of an attenuation in terms of effect size, or C index. But still, I think we had some pretty strong results in terms of the net reclassification index, particularly when it comes to continuous. So if you're looking not necessarily just at thresholds and who passes thresholds, which I'll go over in a second, but basically our NRIs for continuous NRIs for polygenic scores were about 10% of cases, future cases, correctly being reclassified upwards in terms of risk, and about 12.5% of non-cases correctly being reduced in terms of their 10-year risk for CVD. And this was pretty good, especially in terms of cases when compared to other sort of very promising biomarkers like C-reactive protein as well. So you can probably see there that 10% for the PRS of cases were up or increased in risk, whereas only 2% or so was the case for CRP. So as I mentioned before, we did a recalibration, which is important in itself. I can explain a little bit more about that later if people are interested in. But basically, the main result is that when you go through and do a targeted risk screening strategy, that is, you go for people who are in an intermediate risk range and you add the PRS there, basically in the 5 to 10% 10-year risk, and you add PRS information on top, you end up, say, per sort of 100,000 individuals screened, you end up increasing the risk classification in terms of people who you'd actually give a statin to by about 357 of those people get increased in risk, and they end up being cases, right? So these are actually cases that get increased in risk and are treated with statins. So multiply that number by 0.2, and you get about 75 CVD cases prevented per 100,000 people per 10 years. So that's, you can sort of extrapolate from there. So that's a general sort of estimate that's based on a primary care population in the UK. And you can add sort of your multiplier there to extrapolate to whatever sort of size you want. But we're talking about, thousands at the population level to 10,000s if you're talking about big countries, that sort of thing. Prevented CVD cases per 10 years. So non-trivial. And really what came out of this, I think, was that really sort of striking to me was that, what looked like modest increases in C statistics really translated to what we think or what we estimate to be, really quite meaningful clinical benefits when paired with statins. Okay, so I sort of mentioned sort of the Polygenic Score Catalog. This is one of the reasons why I was asked. Inevitably get to that at the very end. So I've given you kind of a run-up of polygenic scores, their utility in sort of early childhood and more autoimmune settings, more broadly in terms of highly polygenic diseases like CAD and stroke and their combination. And now I'm going to tell you a little bit about how we facilitate translation of polygenic scores at a much higher level, right? How do we give the community resources and tools and frameworks that they need to translate their PRSs and with transparency and reproducibility. So you've probably seen, if you follow the polygenic score literature, that there's many different ways of constructing polygenic scores. There's different training sets, there's different validation sets. Some people don't do external validation. Some people use different covariates. They have different performance measures. all this sort of thing. So there's no best practice. There's actually very little sort of standard practice. There's not a lot of commonality really in terms of how people do their polygenic score studies. And that means that you can't, based on the publication, compare polygenic scores across studies, right? So it's all done in all these different ways in different data sets. And that means that, it's very difficult to say what's best, right? And you can see that when you start looking, across diseases, whether it's CAD, BMI, type 2 diabetes, breast cancer, et cetera, everyone's got a different way of doing things. And indeed, in some respects, that's actually okay because each one's got a different sort of setting, right? Courses for courses. But even within each disease, within each phenotype, it's a huge amount of heterogeneity. So the other thing that really struck us was that people, probably about, at the time, it was actually more like half, but people just weren't really sharing their polygenic scores either. So you couldn't actually take a published polygenic score and try and reproduce it or test it in your own data. And we thought that this was a problem, right? So people were kind of hoarding their polygenic scores, not reporting them, sort of the reporting metrics were just all over the place. So we started this thing called the Polygenic Score Catalog, pgscatalog.org. You can go there now, you can play around with it. It's really developed rapidly, really what it is it's an open repository of polygenic scores. You can go and download the SNPs and weights for polygenic score and use those in your own data. It's a big sort of entrance criteria to get into, for a published polygenic score to get into the catalog, is that it has to be available. And that, there's a set of performance metrics that are reported and it's externally validated and that sort of thing. So at the moment, we've got about 216 polygenic scores in the PGS catalog for about 100 traits and diseases. And that's based on 87 publications. So over 200 have been curated and considered, but 87 have really met those sort of basic criteria of external validation, polygenic score available, and appropriate sort of reporting and performance metrics, that sort of thing. So you can read our preprint, you can follow us on Twitter. There's a lot going on in that space. So, and again, I'm sort of to some extent repeating myself. The Polygenic Score Catalog, as you can see, has a lot of functionalities. It has various sort of things about publication, about the sort of standardized way of reporting the development method and the performance metrics of the polygenic score. Whether it's hazard ratios, odds ratios, C indices, AUCs, et cetera. There's a PGS scoring file, which is a standardized way that's also harmonized to Build 38 that you can use to apply the polygenic score. And really importantly, we also have a fairly comprehensive cataloging of the cohorts that go into the training and validation of each polygenic score. So this helps protect people against, making the misstep of overfitting and overestimating polygenic score effects when they use their polygenic scores and other people's polygenic scores downstream. So we've got a wide variety of polygenic scores in the catalog. Many, of course, as you can probably see, there's a lot of cancer polygenic scores. There's a lot of hematological measurements and blood cell traits, neurological disorders and cardiovascular disease as well. And all things like IBD, ulcerative colitis, and Crohn's, that sort of thing. And each polygenic score, there's a page for its publication, there's a page for the polygenic score. And on each sort of page for the polygenic, or for the trait, we also give sort of, all the polygenic scores and publications for each trait. And then for the polygenic scores, we also have, you can go to each sort of polygenic score page, you can click on the download link in the upper right, and you can also look at performance metrics. And, we've got a fairly, well, we should probably have that whole thing read, but there is a disclaimer in terms of you can't, if you've got like different, you can't necessarily compare performance metrics, right, against different publications. We haven't gotten into benchmarking yet, we'd like to. But we do also include benchmarking papers in the polygenic score, which I think is a real, really useful thing, right, in terms of when people want to select the most powerful polygenic score for a particular application. And again, as I mentioned, there's training and validation data, lists of the cohorts that were included in both. their ancestries, sample sizes, et cetera, et cetera. So just running out of time a little bit here, but we've also, as we've developed a research program in polygenic scores and developed the catalog as well, we've also teamed up with the ClinGen complex disease working group led by Jen Wojcik and Hannah Wand to develop reporting standards for polygenic scores as well. So I won't go over too much of it here, but this there's a preprint on medRXiv to that extent, and really it's about coming up with a list of, in this sort of publication 33, we've kind of pared it down and reviewed to about 20 or so items which are necessary. It's a minimal set of criteria for reporting polygenic scores, right? And these are basic things, right? Like training sets and external validation performance, and what's the kind of potential clinical application here, what's its use case, what it might be useful for, that sort of thing, the ancestry of the individuals, all of these things that really are meant to help standardize the reportings of polygenic scores and make sort of facilitate their translation as well. It's because if you've got some poor reporting, you can just imagine that no one's really going to want to use it unless they're confident and that they trust that the study's been done to a rigorous standard. Future plans for the Polygenic Score Catalog, we're rapidly increasing curation. This is still manual. It is intensive, but we're also, we're making a lot of progress there. We're implementing machine learning-based literature triage to help comprehensively capture a lot of the polygenic score publications. There's over 1,000 of them at this stage, so a lot to get through. We're also we're planning to combine the PRS-RS, the reporting standards, with the Polygenic Score Catalog to basically assess each PGS against those reporting standards. And as I mentioned, benchmarking is extremely important. It's underdone in the literature. So the Polygenic Score Catalog will be getting into to PGS benchmarking as well, so that we can directly compare polygenic scores for the same trait on the same samples. So in summary, I've presented to you a background of polygenic scores, their sort of application and translation, really sort of stressing, I think, that we're really talking about different sort of use cases here. It is very much horses for courses. We can't really talk about polygenic scores in general. Each sort of disease has a different framework. And also, hopefully trying to take advantage of the unique properties of polygenic scores, that, that they are based on germline DNA, and that, we can use these things early. So we should think about them in terms of the first risk factors, right? And there's, again, it's also worth pointing out that we are looking at common variation here, and there's a lot of people who are thinking that, we really should be combining monogenic and polygenic variation together, and I'm one of those people. So that we can actually, have much broader, genetic risk scores that leverage the full allelic spectrum of disease-related variation. Okay, so I'd like to acknowledge a few people, particularly Gad Abraham, who's done a lot of the polygenic score work, Rodrigo Canavas, especially with JIA, and Sam Lambert, who's been instrumental in getting the polygenic score catalog up and running and usable for the wider community, and it really has sort of taken off. So I very much encourage everyone here to have a look at it, rummage around. There's programmatic access through a REST API, and it's really there to be utilized, and hopefully support everyone's research. So thank you again, and happy to take questions.
Thank you, Mike. This is the great time and the great presentation, and then we are open for the questions. So we have about 5 minutes for the Q&A session. I have one question typed in the chatting window, but actually I cannot understand the question very well. So the question is, are any similar risk scoring done and compared with the similar genetic species that have no such traditional risk factors? So Michael, can you take on the question and then answer?
In terms of genetic species, could you clarify?
So yeah, so that's the part I don't understand. So can you unmute and talk to Mike directly?
There's no name for this person who asked the question. Okay. I think if there's no details, maybe we can.
Move on to the next question.
Okay. The next person who will have a question is Andrew. So Andrew, can you unmute yourself?
Yeah. Hi. You touched on the differences with ancestry and ethnicity, and obviously, like the first solution be getting more and better data. But when we have that, is there a case for like ancestry specific polygenic risk scores for disease, or just curious about that?
Yeah, look, so I think that's something that has to be done, in my opinion. I mean, each ancestry, as far as we can see, has a different mean and variance of a given polygenic score. So, and that's due to allele frequencies and linkage disequilibrium. We are going to need different scores for different ancestries, and that's a major challenge, right? Because where are the boundaries, and how do you make decisions about which score you use in which case is a real sort of challenge. And we do need more data, and the more we can collect, the better we're going to be. And I really don't see any other way around it. We can do sort of fancy things like transfer learning and trying to model the LD and allele frequencies directly, but in the end, we are going to need those non-European data sets to get better scores.
Thank you.
And then we have a second question from Christy Murphy. So Christy, can you unmute yourself?
Yep. Hello. I was wondering about, in the clinic setting, how much weight are you giving the polygenic risk score? Just keeping in mind the limitations, based on, ancestry or ethnic background, how much are these scores, or should they, impact and influence patient care and patient testing?
Yeah, so I mean that's the million-dollar question. I think we're still a ways from how we weigh up all this information, and it is going to be disease specific, and even I mean you could also take this even out of the you could talk about completely healthy individuals who have no risk factors and what they actually do with that information in terms of screening, when they might initiate screening, how intense the screening might be, etc. There's no simple answer. I wish I had even something, an example, like a really confident example to give you, but I don't at this case. There's a lot of studies going on in Boston and elsewhere that are looking specifically at this question. So I think one of the things that we're considering and that is useful in the UK setting is things like NHS Health Check. So each individual goes in at age 40 and does a comprehensive health check. But as you can imagine, some people, maybe they need to go in before age 40. So especially when it comes to cardiovascular disease, we do know that men are at much higher baseline risk than women. And, we've also got a GRS that, as you probably could see from those risk curves, there's a substantial proportion of men who, their risk accumulates a lot quicker than others. So, one of the questions we're asking is, can we capture, or can we stratify men in the population who should come in for the NHS health check early, before age 40, and what that might mean for their CVD screening from thereafter. And initial indications are, well, heck yeah, there's a big proportion of men who should be coming in before age 40, at least in the UK.
Thank you.
Just to pass the 1 o'clock, so so Mike, if you can stay a little bit longer, so I'll be happy to have you and then answer more questions. So yeah, feel free to log off if people have other next meetings. So Leon, any other questions?
Yes, I do have a naive question maybe. So so for what kind of PGS scores are based on the data and you know, and what's the European samples? And I know that it's actually very important to choose data from the PGS database when we try to calculate the PGS score. So and then if we don't have better data, usually we have a bias, you know. So could you actually introduce a little bit more, like how could we choose the population when we try to calculate the PGS score from your database?
Yeah, so so in terms of, so you want to, so if you're using the PGS catalog, we do our best to annotate the cohorts that have gone into the training of the polygenic score, and which also sort of go back to the original GWAS if it's using summary statistics. So to some extent, you can estimate, you know, how similar the ancestries that went into the construction of the polygenic score, and maybe, you know, what proportions, because typically especially when you're talking about CAD, you know, you've got mostly Europeans, but actually a lot of South Asians, African Americans as well. So, you know, you kind of get a sense of, you know, how similar the training sets, right, the data that went into constructing the score are to the application setting, right? So we kind of leave that to the user at the moment, right? And there's really no substitute for looking at, I mean, you kind of have to take, you know, download the scores and try them out, right? Mm-hmm. And see, you know, this particular score, the underlying GWAS had like half South Asians, let's say, and I'm looking at a South Asian ancestry data set, and that score seems to perform better than the one that's based exclusively on Europeans. At this stage, really, that's what we're at. It's very empirical, and you know, I think we're going to be like that for a while now, until we have a really comprehensive benchmarking of the catalog, and then we can start saying, well, okay, here's an external data set, here's all the scores for a particular trait, here's how they perform in the different ancestries of this data set, and then you can kind of try and map on your individual kind of ancestry to those, right? So you get a sense of what the kind of predicted effect, efficiency, whatever it would be, of the PGS would be in your data. Hopefully that answers your question.
Yeah. Thank you so much.
If there's another questions, then I can ask my question, if Mike is okay. So Michael, so what's the SNP heritability of the CVD in general? What's the kind of the narrow sense heritability when you calculate that?
Yeah, so cardiovascular disease is kind of, you know, a bit of a composite. For coronary artery disease, the narrow sense heritability is somewhere between 40 and 60% generally, is what people estimate. So it's not surprising at all in that way. Things like stroke, it really depends. I mean, stroke is a highly heterogeneous phenotype. You probably know the major distinction between ischemic and hemorrhagic. But the heritabilities there are a little bit lower. Is very difficult, especially when you're talking about, like, say, SNP heritability, which is kind of like, you know, also the narrow sense and the SNPs that are on an array. So it is lower there. And certainly, you know, I mean, you probably know that CVD is far less heritable than, say, you know, celiac disease or type 1 diabetes, that sort of thing.
Yeah, that's true. And then I actually want to learn about your thought about the, if we want to actually introduce this, the PGS or the PRS to the clinic, so what's the kind of the threshold for the AUC or the C index to have the clinical validity to introduce to the clinic? So like a 0.7 is a good number to start thinking about it, or even higher?
Well, I think one of the key takeaways from the analyses that we've done is that, you know, the C index can be really misleading. You know, it's a very difficult. It's kind of best for model selection, to be honest. Yeah. I mean, you know, really when you have, you know, this versus that. But when it comes to clinical utility or even, you know, clinical validity, you kind of really need to jump directly to, you know, how many cases am I reclassifying from this risk group to a risk group where I would make a decision and treat someone, right? Mm-hmm. So, and then how many kind of, you know, events would I prevent in doing so? I think it's an open question as to what's a number, what's a threshold. I think we kind of need to think about that in a relative setting. So that's one of the reasons why we compared the PRS in the CVD case to C-reactive protein, which, you know, many people have suggested that it should be included in you know, risk prediction models, and indeed it is in some. But, you know, with PRS, I mean, we're talking about, you know, far more, you know, almost double the hazard ratio, almost double the C index increase. So, you know, and I gave that the number of, you know, we're estimating roughly 72 additional CVD events predicted per 10 years per 100,000. You know, you can extrapolate that out to the US population, whoever you might consider for CVD screening. And, you know, it's a non-trivial number.
Right, right. But one more question I have is because you are located in Australia as well. So the Australian population genome project, like 2 million Australian genomes will be done in next couple of years, if I remember correctly. So are you preparing for the CleanGen polygenic score reporting system for the Australian population genome sequencing project?
I mean, it's early days. I guess, to be honest, I think everyone at the moment is kind of waiting and seeing, you know, what you know, big genome projects and how difficult it's going to be, and what the revised timeframes are going to be. So, yeah, I guess we'll see. But, you know, we're designing this, you know, not just for Australia but for the world. So, you know, hopefully in the next, you know, this year we'll have, you know, the reporting framework, the open catalog, and hopefully soon to follow a set of best practices, and ideally, you know, a PGS curation platform as well as benchmarking. So I think, you know, the next sort of year, there's going to be a lot of very, I think, some very positive steps, some advances, you know, really sort of solid foundation to translate polygenic scores.
Mm-hmm. Yeah, I think, yeah, so yeah, thank you for staying like a 10 minutes longer than as we before planned. So thank you very much for your nice talk and great Q&A session. So Liang, if there's any other questions?
No, I just want to thank you, Mike. It was a very interesting talk. And I also want to announce that we actually have a lot of seminars on September 8th. So if we don't have any more questions, we'll see you guys. Thank you so much for joining us.
Okay. Thank you very much, Mike.
Okay. Thank you, Mike. If you want to email me any questions or anything, I know a lot of people hang out afterwards. You know, feel free. My email's on the internet.
Great. So Mike, actually I have one technical question. So have you ever compared the medRXiv with like the BASAR or SBASAR by the Vicious group?
Yeah, I mean, so medRXiv.
I'm asking the question because the comparison table you showed us during your presentation didn't include that the BASAR or the SBASAR. So I'm asking that now.
So we've there's a method, there's a bake-off that's being prepared, I suppose.
I see.
In terms of PGSs, its methodologies. It's a little bit better. So, I mean, you probably know that, well, if you're looking at a score of scores, that, you know, it's something that can kind of suck in everything, right?
Yeah.
So in terms of, you know, depending on summary statistics or individual level data, you know, you're really limited at that level, whereas a metascoring framework is really able to draw on a whole lot of different sources to get its predictive power. And you just need to, you know, estimate, you know, the weights to how to combine all of those. So the comparison itself is actually non-trivial to do something that's fair and that sort of thing. So
yeah,
at the moment, it looks like it's doing a little bit better in the sort of fairest to the fair comparisons. But I may have to get back to you on that one as well, because I haven't looked at the latest.
So what's the journal that you are submitting to publish the catalog, the Polygenic catalog? So I saw that on the medRXiv, but it looks like you are submitting to some the journal already.
So it's in revision at Nature Genetics.
Oh, nice.
Yeah, we'll see. You know, hopefully it gets in. I won't jinx anything.
Great. Okay, so yeah, thank you so much, and then hope to talk to you more about the PRS at the Boston Children's. We are doing some like a genotyping and larger scale genotyping of the pediatric cohort as well. So we want to apply some the polygenic scoring, but it will be a little challenging, especially for the pediatric population.
So we should, yeah, maybe we should catch up after this at some stage, because there's a lot of discussion at biomedical centers, which, you know, kind of like these conglomerations of hospitals and NHS trusts. They're planning and in some cases doing the same thing on their hospital populations. So a lot of people are moving in this direction.
Exactly.
Yeah. So
Great.
yeah, it'd be good to catch up on that, see what the opportunities might be.
Yeah, that will be perfect. Thank you so much.
Great.
Bye.
Thanks, everyone.
Thank you. Bye.
Bye-bye.